Olá a todos, sejam todos bem-vindos à nossa tradicionalíssima conferência de quartas-feiras, agora oficialmente rebatizada como Conferência Professor Marcello Barcinski, que vale lembrar, essas conferências compõem a disciplina BFB 704, que é a Informação Científica, a disciplina da pós-graduação. O nosso ilustre convidado do dia é o Dr. Georg Fantner, da École Polytechnique Fédérale de Lausanne, EPFL, motivo pelo qual agora eu vou fazer a apresentação dele, né, em inglês. So, hi, Dr. Georg. Well, Dr. Georg received his Ph.D. degree in physics from the University of California, Santa Barbara, in 2006. He was an Erwin Schrödinger Fellow at the Massachusetts Institute of Technology, Cambridge, for some time before joining the faculty at the École Polytechnique Fédérale Lausanne in Switzerland, where he works as an associate professor. There, he heads the laboratory for Bio and Nano-Instrumentation, the Interfaculty Institute for Bioengineering, and the Microengineering Institute. He has authored many internationally recognized papers and book chapters. His current research interests include developing next-generation nanoscale measurement technologies for the life sciences. Well, he also leads a project called InCell, which uses atomic force microscopy to image events at the nanoscale inside living cells. So, dear Dr. Georg, the Chagas Filho Biophysics Institute thanks you for joining us today. Please feel free to start your presentation whenever you're ready.
All right, well, thank you very much for the invitation. It's great to have this opportunity to speak to you. Obviously, it would be much nicer to do it in person, and I very fondly remember previous visits that I had to your institute. And so it's really great for me to have the opportunity to give you an update on the work that we do in the lab. Thank you for the introduction. I don't have to say much more about the background of what we do, except for maybe explaining a little bit the kind of presentation I'm going to give today. I want to give you a very broad view of the kind of research that we do in the lab, and that's why I titled the presentation Challenges and Opportunities in Scanning Probe Microscopy, which really could mean anything. But it does point towards the fact of what it is we focus on and what I like to do, which is try to build new techniques, invent new measurement technologies to enhance the capabilities of what we can measure in material science and life science. And most of the techniques that we are developing are around the technique of scanning probe microscopy. Oh, let me see if this is, why is this not working now? Yeah, no. Did the slide change? I'm just going to assume it changed.
Yes, sir. Yes.
Perfect.
It's changed.
So we're working in scanning probe microscopy, and one of the most used scanning probe microscopy techniques is atomic force microscopy. And it's a microscopy technique that is very well suited for imaging biological processes and biological samples at the nanometer scale. And that's because it's one of the few high-resolution measurement techniques that can actually image biological samples in their close to native environments. So you can image with atomic force microscopy in solution, in buffers, in growth solutions. So you really can try to keep your sample alive and follow a biological process. And that's really been the theme of my lab ever since I started it many years ago at EPFL. Our goal was to not just get high resolution pictures of the structure of the biology, but actually see how that structure changes. Because living things, they change. That's at the nature, at the core of life is that it changes. And so we have been applying a lot of commercial atomic force microscopy methods to imaging cell growth, cell division. Here I have some examples of some time-lapse imaging where we were doing very long-term imaging of bacterial cell growth and cell division. But very often we got to the limit of what we could resolve, because atomic force microscopy takes relatively long to take an image, and the smaller we get in biology, the faster things change. Here, for example, we were working on doing a little bit higher speed atomic force microscopy combined with super resolution optical microscopy. And we were able to image living cells, topography, which you see here. Let me see if I can actually get a pointer, which you can see here. And then at the same time, also image the focal adhesion points and how they change over time. However, we noticed that the speed of conventional or commercial systems just was not sufficient to track the biological questions that we were interested in. So we had to start thinking about how can we improve scanning probe microscopy in general and AFM in particular. And those of you who know atomic force microscopy and who have been working in the field for a while, they know that there is an almost endless number of different ways to use the atomic force microscope. There's many different modes, there's many different measurement modalities, and all of them have their own individual challenges and opportunities. But when we talk about improving these, all of them have very similar bottlenecks that we can build on. One bottleneck for all scanning probe microscopes is the speed. And so I will actually dedicate quite a lot of my talk to the topic of speed. But the other thing that is becoming more and more important is to not just use the AFM as an imaging tool that gives you the structure, but as a measurement tool to really extract quantitative information about the sample that you're imaging. Another topic that is being worked on in my lab a lot and also has been getting traction in the community is the compatibility of scanning probe microscopes with other techniques, because there is a lot of great things that we couldn't do with AFM, but there's a lot of things that we cannot do with AFM. And so the combination of imaging techniques, other imaging techniques with atomic force microscopy is currently a very big and active field of research. And then we're always looking for different ways to improve what we can measure. So the versatility of how you can use the same instrument for very, very different purposes is also something that we always look into, and we try to find new ways to use the existing techniques. But like I said, I will first focus a little bit on the speed. And just to give you a bit of a feeling of what I mean by AFM imaging speed is low, because we are scanning with a cantilever probe the surface of a sample line by line, we have to do that in sequence. So if we scan with a normal atomic force microscope, let's say 1 line per second, then it takes many, many minutes to get a high resolution image. To overcome that, there's been for the last, well, close to 20 years now, people have been working hard on improving the speed, and there's actually quite some amazing work that has been done in the field that's called high-speed atomic force microscopy, which is primarily focused at looking at molecular self-assemblies, molecular motors, and changes really at the very much the nanometer scale. So what required to make that step from the general AFM, as the ones that you probably have in your laboratories, and the high-speed AFM? Well, one of the key parts of the atomic force microscope is the atomic force microscope cantilever. That is the sensor. The tip is where the magic happens. The tip interacts with the sample and extracts the topography. Everything else is just there to read out the cantilever deflection or the cantilever motion and then react to that motion. But the whole measurement starts at the cantilever. So if we want to optimize the AFM cantilever, they have the AFM speed, we have to optimize the cantilever as the first step. So one of the things, we have to increase the resonance frequency. This goes very much hand in hand with increasing the bandwidth of the cantilever, and I'll get to that in a second why. And we also have to improve the way in which we can actuate the cantilever. We have to make sure that we actually can probe the sample fast enough so that we can respond to changes in topography rapidly. And then there's other things that are important that people are working on, but not so much in my lab, which is increasing aspect ratio of tips, reduce the tip radius. Those things are very, very important to increase the resolution and the ability to also measure very complicated structures. Now, I know that there's been there are a few people here in the audience that have been doing AFM for a while. I mean, Gilberto is here. And I don't know, Gilberto, if you can remember in the early days of AFM, when we were all making our cantilevers by bending little pieces of wire and putting diamond tips at the end and little reflective mirrors on the back. Well, fortunately, we've come a long way from that, because these were relatively large cantilevers and they were good for static measurements, but they were not very good for doing dynamic measurements at high speed. So people have worked now for many, many years on improving AFM cantilevers in certain areas, certain aspects. I already mentioned the spatial resolution, which is primarily tip radius and tip aspect ratio. But for high-speed AFM, way more important is the sensitivity and the bandwidth. The idea here is how quickly can the cantilever detect the surface and with which sensitivity. And another area where people are working on improving cantilevers is to increase their compatibility with different measurement environments, and in general, putting more intelligence inside the cantilever, so that it's not just a passive mechanical force sensor, but actually putting in measurement electronics, actuation electronics, to put a little bit more smarts into the measurements. And I will have some examples later on as well. But first, I want to focus on the speed of the cantilever. Why is this always coming in? Well, the cantilever has to physically interact with the surface. And in most measurements that we do in biology, we're working on very soft samples. We're working on fragile samples. So we cannot have the cantilever sitting on the surface, which is its called contact mode, and drag it over the surface, because we would be distorting or even destroying the samples. So that's why people are primarily working in what's called tapping mode or amplitude modulating mode, where the cantilever is excited at its resonance frequency and only intermittently touches the surface. And depending on how close the cantilever is to the surface, the amplitude of the oscillation changes. And that is what we're using as the signal to feed back the AFM topography. So that's the variable that we're detecting, the amplitude of oscillation. Now, if you think back into your basic physics course, if you were talking about simple harmonic oscillators that were oscillating in a certain confined area, then you know that you get a certain amplitude out of that. Now, if you change that confinement, the amplitude does not automatically or not directly go to the new steady state. It takes a certain number of cycles for the oscillator to reach the new amplitude. That's very, very basic physics, because the energy that the cantilever requires to oscillate at a higher amplitude first has to get into the cantilever. And what that means now for AFM imaging is that if we are oscillating in tapping mode along, and then we're getting to a change in topography, the cantilever initially does not actually change too much in the amplitude. That's because this change in boundary condition, this change in topography, only will be detected once the cantilever has obtained enough additional energy to reach the new steady state amplitude. And that time during which we're having this ring up, we are blind to the surface. So this time determines how quickly we can scan and how quickly we can take our image. That is the easy solution for that. We can just make the cantilever oscillate faster. If you have a higher resonance frequency and it takes the same number of cycles to reach the new steady state, then if you have this new higher resonance frequency, you will detect the surface faster. And this is the way that most people are working when they're working in high-speed atomic force microscopy, is increase the resonance frequency by making the cantilever smaller. The smaller the cantilever, the smaller the mass, and at the same spring constant, the resonance frequency is going to be higher. We can characterize the speed with which we can actually detect the surface topography with a bandwidth. So how quick or what changes can we still detect as they're coming, at which frequency can we still detect them with the cantilever when it's oscillating. And this bandwidth is proportional to the resonance frequency, like I explained here. And it's also the Q factor, because the Q factor of the oscillator tells us how many cycles the cantilever has to oscillate in order to get to the new steady state. And this Q factor has to do with the amount of damping that is inside the cantilever. So in theory, if we could control this Q, then we could also achieve a high bandwidth by keeping the resonance frequency constant. So we could make potentially a cantilever that has the same resonance frequency, but a lower Q factor, and we would get the same speed with which we detect the topography. And ideally, of course, we would do both. We would make a very small cantilever that has a high Q factor, that has a low Q factor, and therefore would only require very few cycles to find the new steady state, and but at the same time go through those cycles very quickly. Now, the way to go there to get that is to start working on this Q factor. And one thing that has been overlooked for a long time was the material of the cantilever. Everybody was optimizing the shape, but the material was pretty much always very standard microfabrication materials, silicon or silicon nitride, as we know them from the semiconductor industry. But if we actually look what material parameters determine how fast such a cantilever reacts, so what determines the resonance frequency and the Q factor, we can plot on the y-axis the mechanical properties that determine the resonance frequency, which is Young's modulus and the density. And on the x-axis, we plot the mechanical properties that determines the Q factor. So the property that determines the Q factor is the intrinsic loss coefficient of the material. And so all of these lines that are here, these dotted lines, they all have equivalent bandwidth, so to speak, from a materials perspective. So if you're down here, if your material sits down here, your cantilever is going to be very slow, whereas if your material sits up here, it's going to be very high. And if we look where the normal materials are that we make cantilever out of, silicon, silicon nitride, some people make them out of silicon oxide or quartz, we are in this regime here. But there's another group of materials that can be used for microfabrication that are 2 or 3 orders of magnitude potentially faster in material to make cantilevers out of. And so we've picked that up a couple of years ago, And we've made cantilevers out of SU8, and we were able to increase the speed by a factor of 100 by just changing the material properties and optimizing the geometry. I decided not to give you too many details about this because it's really more engineering and material science, but rather I want to take this now and show where we went from there. So with this tapping mode, we can, and these polymer cantilevers, we can get 100 times speed improvement during the imaging. But there actually is another way, and that other way has to do with trying to no longer need this Q at all, this Q factor. This bandwidth equation here is valid for when you are exciting the cantilever at its resonance, when the oscillator is resonating. But what happens if we use the cantilever not at its resonance frequency, but at a frequency far away from the resonance frequency? Well, that is what we call off-resonance tapping. In off-resonance tapping, instead of shaking the cantilever at its resonance frequency, we are actually moving it up and down in a controlled fashion and touch the sample, come back up, and retract it. And we do that for every pixel at least one time. The frequency with which we do this is generally much, much lower than the resonance frequency. And there's actually a number of different commercial implementations of this off-resonance mode. You may know it as peak force tapping, or you may know it as QI mode or hybrid mode. All of those work in a similar way. And they have the very big advantage that here now, you are getting one interaction curve per pixel, irrespective of the Q factor of the cantilever, at least to first order. The other advantage is that you're getting a not just a topography height, but you're actually getting for every pixel such an interaction curve, so the deflection as a function of time. And if we do a little bit of coordinate transformations, we can actually get a force distance curve out of that. So we get a force indentation curve for every pixel of the image. And that is great because we can use it to extract mechanical properties, which we will see a little bit later. Now, the issue here is this motion of the cantilever up and down happens with a piezo actuator. And you have to move either the whole sample up and down or the whole cantilever up and down. And you're moving really a lot of mass, which really slows it down. So off-resonance AFM modes tend to be even slower than tapping mode is. So it's not really helping us in that respect. So what we've done now is we've said, okay, well, all we need to do is we need to modulate the tip sample interaction. And instead of doing that by going up and down mechanically with the piezo, is what we did is we added a second laser beam into our microscope. And that second laser beam is actually focused at the base of the cantilever. The first laser beam sits at the tip of the cantilever, like every other AFM has, to read out the deflection. But the second one is here focused at the base. And instead of shining a constant light on it, we pulse the light that we are putting on here. And so we are locally heating with the laser the cantilever. And because the cantilever consists of different materials, so we have the base material, in this case silicon nitride, and on top a metal layer, in this case chrome and gold. When there's a local increase in temperature, these materials expand differently, and we get different, we get this bimorph effect, which will actually bend the cantilever down. And the big advantage now is that we can do this much, much faster because we only need to move this very, very small cantilever and no longer the whole mechanical structure. So with that, we can now drastically increase the speed of the off-resonance operation. We call this photothermal off-resonance mode or port mode. In order to do that, we need to have very special cantilevers. We have cantilevers that are both high in bandwidth and small, but also very good in the photothermal excitation and the bimorph characteristics. So we have developed this process to make very, very small custom cantilevers where we optimize both the material properties of the material and of the coatings to get very high amplitudes and very high speeds with this laser drive. Now, of course, we also need the way to drive this laser. And so we've built a modification to our AFMs, where in addition to the readout laser, which you have in most AFMs, we've now added an additional laser, which also is focused through the same objective lens, but now on the base of the cantilever. And here's a cross-sectional drawing of the setup. This is actually a plug-in replacement to the Vico or Bruker multimode AFMs. So if that's one of the microscopes that you have, you can just take the normal AFM head off and put this head on, and it's actually plug-in compatible. And with that, we can now operate this off-resonance mode instead of at 2 kilohertz, which is a typical off-resonance frequency that you can get with a commercial instrument. We can go to several hundreds of kilohertz, and the most recent data by our student is even up to 1 megahertz in this off-resonance mode. So before I show you the data and the kind of research that we can do with that, I do want to spend a little bit of time on a concept that's very, very dear to my heart. I think we have some audio in the background.
Yeah, I'm trying to yeah, I'm trying to mute her.
O valor é muito alto, a gente trabalha de graça, sabe? A gente faz avaliação gratuita de.
Well, anyways, I'll just speak a little louder.
No. Sorry, we are.
No worries.
Where's Christina here? Here. Sorry.
So one of the things that's very important for my lab is not only that we develop new methodologies and new measurement modes, because if we can do it in the lab, that's great, and we will publish some papers about it, but it's not really helping science move forward. And that's why years ago already, we have decided that everything that we design and we develop should be made available directly to the scientific community in the form of open hardware. And so we have started projects where we actively try to help people build copies of the microscopes that we have built and the techniques that we've developed also for themselves. So here I want to show you just the open high-speed atomic force microscope project that we have. We have also other projects, open structural illumination microscopy, etc. But this is by far the one that has gotten the most interest and the most adoption. So what we do here is that we organize workshops where people can actually come to our lab and build copies of what it is that we've designed and we've built. We prepare first all the parts. People come, they get instruction on how to build it, how to test it, how to repair it if necessary. And then after the workshop, they can take this microscope home to their own labs and do research with them. And so far we've had quite a few successful workshops. We try to have one about once a year. Unfortunately, COVID was a bit of a hiccup there, so we haven't had the workshops regularly in recent years, but we just this year started to pick them up again. And so they're about 1 week workshop, depending on how many parts people want to make. And it's small groups, it's very intense training. So for, let's say, 4 participants normally per workshop, we have at least 2 to 4, so to speak, instructors that really help people build and also explain why the instrument is built in a certain way. So if this is something that you're interested in, please let me know. My email address is actually somewhere at the beginning of the slide, because we're planning to do at least one more this year. And the cost of the workshop is purely the cost of the parts. So we're not making any money off of this. This is really the idea is that we want to enable people to not just read the papers, but actually have also access to the capabilities then afterwards in the lab for their own research. So what are those capabilities? What can we do with, for example, this high-speed open, open high-speed AFM? It's a tool that is particularly well suited for studying molecular self-assembly, molecular motors, and generally the dynamics of reactions. And here I have one example that is work that we've done with our good colleague Pierre Guernsey at EPFL. He is in the Swiss Cancer Institute, and he does research on centrioles, and particularly how centrioles assemble and how they maintain their ninefold symmetry, and how the cells generally make sure that after each cell cycle, there's exactly the right amount of centrioles. And at the center, so I don't know, I'm not going to give you an introduction to centriole biology because I'm not the right person for that, but just to show you there are really, really interesting structures that consist of a core protein, which is called the SAS6 protein, which is actually a homodimer. So there's two molecules that are forming a helix and then has a head group. So we have a tail and a head group. And then 9 of those homodimers self-assemble into a wheel, the cartwheel of ninefold symmetry. Multiple of these cartwheels stack up, and around those then microtubules assemble, which form the centrioles. And so the structure is very well known from electron microscopy. It's also possible to image these formed cartwheels with the atomic force microscope. But what was not known is why it's exactly 9, and how does the cell control that it's exactly a ninefold symmetry, because this ninefold symmetry is very, very well conserved all the way through evolution. So the question is, how do these homodimers eventually assemble into the cartwheel formation? And what is the reaction kinetics of this reaction? And so we did some experiments together with Pierre's group, where we tried to look at this. And I'm just going to start the video, and I hope it actually plays well for you. And you see here the monomers coming in. You see the tails and the little head groups. And as more and more monomers come in, so we're injecting more and more, the cells, the monomers assemble, and they actually form these little centriole rings. And we had expected when we were doing this experiment that it would just be one ring after the other, one monomer after the other until we have a ninefold symmetry, and then it would close. But in fact, that's actually only one way these rings can form. So here we have a cutout where actually the addition is, let's say, homodimer after homodimer. But there's also other reaction mechanisms like the one here in the center, where we have two partially formed rings first form, then they find each other. They find each other in the wrong way. They recognize this is not the energetically most favorable way to do it, and they actually have self-correcting forms. Or here in this case, we have first formed a spiral structure, which then afterwards folds back over and creates the ring. This was now the first time that we could actually look at this, but because we now have not just static structure data, but we have movies, and we have many, many movies of many, many, many molecules, we can now do machine learning on these images, do single particle tracking, and actually calculate reaction kinetics on the single molecule level. So we get single molecule data, because we have so many, we can back calculate now reaction kinetics as if this were an ensemble process, but obviously it's not, it's a single molecule process. And that makes this technique very, very interesting, because we now have the ability to track individual molecules. And now we can actually try to ask more fundamental questions. What determines the assembly efficiency? How do mechanical properties of the molecules, how do binding constant, how do binding forces interact the final structure that a molecular assembly is going to have? And this works great with, it's very important for doing it with biological molecules, but biological molecules are very difficult to tune in terms of these mechanical structures, the chemical interactions, and generally the shapes. So we turned to another colleague, Professor Matthew Bastings at EPFL, who is an expert in DNA nanotechnology. And so we engineered custom molecules that have different binding affinities, have different sizes, different flexibilities. And now we are looking at how these DNA fragments interact to form these macromolecular larger assemblies. And the first molecule that we started with were DNA tripods, and they were made through DNA nanotechnology, DNA folding. So we have a couple of staple strands that form, and if you nail them properly together, you can form these tripods that have here blunt ends at the end and have this trimeric symmetry. And if we put many of them together and energetically it's favorable, they will assemble into such a hexagonal structure. And depending on how strong this blunt end stacking interaction is, how flexible the individual arms are, how long these arms are, you will get different formations, different defect correction mechanisms. And that's what we're currently studying with this high-speed AFM. This was our very first paper that we published back in 2019, more of a proof of principle that we can record larger areas also with this, the same high speed, and looking at the individual molecules as they assemble, and also see individual defect corrections. We can then again use machine learning to actually identify what kind of structures these are making. And so the green ones are the correct six-fold symmetries in the hexagons, and then blue are 5 and red are 7. And you see that as a function of time, we actually get correction mechanisms within this self-assembly. And currently we're working on expanding that really to explore the full, the phase diagram in terms of binding energies, lengths, and flexibilities to really study the fundamental biophysics of such molecular self-assemblies. So this is my part about the speed, and I can see that I've been talking already quite a while, so I have to indeed also not just speed up with my AFM, but also speed up with my talking. So I will do that. And I move to the second part of where we're working, and that is increase the information content. All of this, all of the work that I've shown you so far was just looking at images, looking at the topography in a very classical way to use the AFM. But because of this off-resonance mode that I explained earlier, we are getting, for each point in the image, de facto, a force curve. And so this is, we get the deflection as a function of time, which we then can afterwards turn into a deflection as a function of z position, which then is a little miniature indentation curve at every point. And now we can use contact mechanics to get mechanical properties out of this. So we can get stiffness from the slope, we can get adhesion, we can get energy dissipation, we can get stiffness of the whole sample. And so we get different kinds of images for each topography image. So we now really have a multi-parametric imaging method. And this again is something that you can commercially buy. It's something that people probably, or you also have in your institute. But if you're using that, you probably noticed that you have to be really very slow. You have to go half a hertz or so to be able to get proper mechanical properties. And so that's, again, the difficulty for us. We want to look at dynamics, not just in life science, but also in material science. So when we're looking at healing materials, self-healing materials as they undergo phase transitions, we need to get different, we need to get the temporal resolution. And so a very talented PhD student, Santiago, in the lab has built a new microscope, which is our latest and greatest high-speed tip scanning AFM that is actually able to measure these mechanical properties now at rates of up to 100 kilohertz pixel rate, and he can get images in less than a second and still get the mechanical properties. So this is now the next instrumentation imaging area that we are working on in the lab. And this is actually also important because it's an entry into more material science work. And my lab is the Bio and Nano-Instrumentation, so we also are working with about half the lab in more material science applications, in clean room applications. And this is where AFM is really not used to its full potential yet. If you look at material sciences and definitely in microfabrication, AFM is very used in a very basic fashion just to image topography of flat samples. And that really is a shame because AFM has a great opportunity in that it can be very easily combined with other methods. I already showed very early a classical combination of AFM with optical microscopy, but there between AFM and optical microscopy, there is a big mismatch in the addressable volumes and imaging fields and resolutions that can be achieved. What would be a much better match in terms of the type of features that one can image is the combination of AFM with electron microscopy, in particular scanning electron microscopy. Or even better, it could be combined with focused ion beam microscopy so that we can get AFM mechanical properties, topography contrast, SEM we can use for imaging materials contrast, for example, through EDX measurements, and focused ion beam can be used to actively modify the sample and look at those changes. And so several years ago, we started building these machines that can be put into a dual beam SEM FIP. And so the AFM has to be very small, has to be easy to integrate. And most importantly, it has to give access to those beams directly to the cantilever, because we not only want to image the same area, but we actually want to image the same area at the same time. And that was a tough challenge, because normally we had these laser beams here and lots of optics. And all of that space above the cantilever is occupied by the laser deflection. And so it was not possible to add that to a microscope where the top is used for the electron gun and the ion gun. And the solution for that is actually moving away from the standard sort of like passive cantilevers that are really just mechanical structures to start integrating more intelligent structures. So actually bring self-sensing electronics into the cantilever. So replace all of this optical detection by microelectronic devices that are built inside the cantilever. And I don't want to spend too much time on this, just a little bit. So what we're doing here is we're adding little semiconductor strain gauges directly inside the cantilever, which we then can read out through a Wheatstone bridge. And as the cantilever bends, there's a strain in the cantilever here at the base, which compresses the strain gauge, and we can measure it electronically, the change of the deflection. In the same way as we have to replace the optical lever deflection, we also have to replace the optical actuation, so the photothermal actuation. And we're doing that by building little microheaters into the cantilever so that we can electrically heat the cantilever and create the actuation that we need for the high speed of resonance tapping. And here I'm just showing a very brief view of one of those cantilevers that are microfabricated at EPFL. It's a completely new microfabrication process, which actually is a combination of traditional silicon nitride and the polymer processes that I introduced earlier in the talk. So these are not just intelligent, but also very fast cantilevers. And you see here the semiconductor strain gauges built in. Here we see the little microheaters. And with that now, we can do AFM imaging without any of the optics. And that opens up a great opportunity. So here are just some examples of experiments that we've done. Here, there is a healable polymer sample that we're imaging, and we're getting the SEM image, but we're also getting the mechanical topography from the AFM, the stiffness, adhesion, and the dissipation. So this is really exciting for us because now we can not only look at the structure, but also the function of the materials. Because we have this integrated not just in an electron microscope, but actually in a dual beam, so we also have the focused ion beam, we can use the AFM to image and the focused ion beam to slice the sample. And then we can image at the same spot. We can image multiple slices, and then we can do a 3D tomography through the sample, but with AFM reflection. These cantilevers are also, like I said, we're trying to put as much smarts in it as we can. So we can also do magnetic force microscopy. We're applying this to these magnetic crystals for single monopole magnetic materials. It's a whole other story. And here, this is one of my favorite project that we've done, is we're actually looking at a piece of industrial rubber, which is a combination of rubber and carbon. And as we're imaging, first we're imaging with the AFM, the mechanical properties, then we're cutting with the focused ion beam through the sample, and then we can reconstruct the 3D density map of the industrial rubber. So this is just a comment that I wanted to make about applying these techniques that we initially developed for biology. They're also very, very useful in material science. And now I want to get to the last part of my presentation, and I see that I'm really already coming to the end, so I will try to keep it brief, but it's also one of the most exciting parts that we're currently working on. And that is trying to do different things with scanning probe than the traditional AFM that's being used in biology. Because the problem when we're trying to look at living samples, especially eukaryotic cells, is that we still have to interact physically with the sample. So we have a mechanical contact between the cantilever tip and the sample, and what we're actually measuring is not the surface of the cell, but the distorted surface of the cell. We're actually bending the membrane, so we're deforming it, and most of the times the cells don't like it, and we don't get a faithful reproduction of the cell surface. Now, fortunately, many years ago, very soon after the invention of the atomic force microscope, there are people who invented a variant of scanning probe microscope, the scanning ion conductance microscope. And the beauty of that microscope is that it no longer uses a cantilever to detect the surface topography, but it actually detects, it uses a electrophysiology pipette, a glass pipette, to measure the surface of the sample. This is the same concept, but it's slightly better rendered. So what we have is we replace the AFM cantilever with a glass nanopipette. This is just a quartz physiology pipette pulled to a point to have a very, very small opening. And we put an electrode inside at a very conventional electrophysiology setup. Now, if we are far away with our pipette from the surface, we have the surface here, our cells in an electrolyte solution, in this case, it's just growth medium, and we have the electrode inside the pipette. We have a current amplifier and a counter electrode. When we apply a voltage, we create a closed circuit, voltage source, and we have a couple of resistances in this circuit that determine the current. Now, if we are far away from the surface, the current is high. But the closer we get to the surface, if you think of this as a little hole that ions can flow out of, when we get closer and closer to the surface, the gap between this hole and the surface gets smaller. And so it reduces the area that the ions can flow through, and the current will drop. And this is what we measure. The closer we get to the surface, the more the current drops. So now we have a non-contact proximity sensor. We can tell where the surface is by looking at this ion curve without ever having touched the surface. And if we do this now at every point in the image, then we have a non-contact way to image the topography of the cells. And this is great because now we can address not just the stiff kind of cells like bone cells or muscle cells that we can do with AFM, but really more the interesting, very, very soft cells, stem cells, neurons that are just simply not addressable by AFM because they just get completely broken. And here's just a little sketch animation of how this works. So you have the pipette coming down, you go down to a certain set point every time, and then you record how far you had to get down to get to the set point. And that distance is then the envelope of the sample, and you can reconstruct the topography. That's a great technique. It's been around for a long, long time, but it's never been particularly successful in science because it's even slower than an AFM. A good SICM measurement takes about an hour per image, which really is not good if we want to look at the dynamics, which is the whole goal that I started this presentation with. So we decided to just apply all the things that we've learned during the development of high-speed AFM in terms of mechanics, electronics, control engineering, and all of these other aspects to build a microscope that can do this imaging, but again, 100 to 200 times faster than the traditional microscopes. This is a rendering, a cross-section rendering of our new instrument. And you see here the glass pipette. Up here you have the piezo actuators that are doing the hopping. All of the electronics goes here through the glass pipette and it actually sits up here. And then we had to develop many other things. I'm not going to go into this, but there's a lot of electronics development. Actually, some custom IC manufacturing was involved, a lot of software programming. But we've been able to increase the imaging speed of the scanning ion conductance microscope by more than 2 orders of magnitude. And that now lets us do really exciting things. First of all, it lets us get images, scanning ion conductance microscopy images of living cells at relatively large scales and reasonable time scales. So they're several minutes to take an image. But then as we get closer and closer, we can increase the speed more and more. And this here is now, sorry, it's covered by the image, but this is now taking at 13 seconds per image. But the current system that we have goes down to 2 images per second. So this is now a speed with which we can very well look at processes that are happening at the cell membrane, endocytosis, proteins drifting. The goal is to start looking at cell membrane transports and things like this. So here's an example of such an experiment that we've done. Here we're looking at melanoma cells from a collaborator of ours. And I don't want to go into the science here at all. I just wanted to show you the kind of images that we're now getting. So here we're starting with one cell. The cell is growing. It's in 37 degrees. It has all the nutrients that it wants. It has the right CO2 concentration. And you see it's actually quite happy. And it's proliferating. And over time, we can see the changes in the morphology. And here at this point, we're actually adding a drug called forskolin. And you start to see changes in the morphology of the cells. It actually starts to create these long protrusions. It interacts with other cells, and you also see the responses to the drugs on the cell membrane. So here you see these we presume are the secretion of extracellular vesicles, but we're still in the process of nailing this down. So this is now a dynamic technique to image live cells without distorting them, and actually we're able to do this over long periods of time. all of this we did because we had moved away from the AFM cantilever to glass pipettes. And now glass pipettes in biology are nothing new. We actually took a lot of knowledge from the people doing electrophysiology, where people are using little glass pipettes for ion current measurements. They've been doing that for a long, long time, and getting Nobel Prizes all over the place. And so it's a very, very mature field. Out of this electrophysiology field and the measurement of single ion currents, single ion channels, there's been a different technological field that emerged, and that's the field of nanopore sensing. I assume you've heard of this. The idea behind nanopore sensing is that if you have, for example, a biological ion channel, a biological pore, then if you have a pore that is inside a membrane that separates two chambers, and there's electrodes in each chamber with a bias voltage and a current meter, then you measure a certain current as there is no molecule going through, but because there is a current from one electrode to the next, and the molecule is charged, it gets fed through the single ion channel and blocks the channel, and you see a change in the current as a function of time. And this is being used very successfully for DNA sequencing. You can actually buy a little USB sticks from Oxford Nano to measure the DNA sequence on a single molecule level. And it's a really great technology. I can only recommend you try it. The problem is, come on. Oh, no, there. The problem still with this technology is that it's relying on biological nanopores. Although the technology is very well advanced, it requires a lot of biochemistry and clean working to have these biological nanopores there. The other issue is that they work primarily with single-stranded DNA, and you can't use it, for example, for double-stranded DNA. And that's why people have started to look at different types of nanopores. They're actually trying to use the original glass nanopores directly as a sequencing mechanism. And so this is work by Ulrich Kaiser, who has used the same kind of glass nanopores that we are using for scanning out conductance microscopy. He's using them for single molecule detection. And for example, he has generated these DNA strands with these little dumbbell sides on it, which are molecular rulers with protrusions spaced at dedicated points. And he can actually detect these things going through the nanopore. The challenge here is that if you're doing that, the molecules, they translate through this nanopore at an uncontrolled velocity. Whatever the bias voltage drags them through, that's the speed that's happening. And once the molecule is through, the molecule is gone. So this leads to a not very good signal-to-noise ratio. We can detect the molecules, but we can say nothing about the structure of the molecules. And here, again, two very talented PhD students, Samuel Laito and Vytautas Novikas, have developed a different way of doing such single molecule translocations, which we now call scanning out conductance spectroscopy. And the idea is here that instead of letting the molecule flow freely through the nanopore, one side is tethered to the glass cover slip, and the other side is free and gets sucked into the pipette. And as it gets sucked into the pipette, now we have full control over the molecule. So the molecule is inside the pipette, and it translates at the speed at which we are moving the glass nanopore, which is a controlled motion that we can control with the piezo, and we can move it up, down as many times as we wanted to. So this is how the experiment works. So we have a scanning out conductance microscopy scanning ion conductance microscope on top of an inverted optical microscope. We functionalize the surface to tether down the DNA. And here we have an experiment. We were doing the binding of dCas9 to double-stranded DNA. And so we're coming in. First, the molecule is outside. We're coming closer. The molecule gets sucked into this into the pipette. And as we are moving the pipette further away, we're starting to feel the influence of the bound dCas9. So here, this peak is due to the dCas9 being bound at that particular position of the DNA. And then as you pull it further, now here it's completely pulled out. And this is now giving us a lot of opportunities for the single molecule detection and primarily for single molecule biophysics. Because if we compare the free translocation through standard nanopores with this kind of controlled translocation, we have a number of different advantages. One of them is that while the other techniques are measuring the current through the nanopore as a function of time, we actually measure the current through the nanopore as a function of distance. So here you have a comparison of these molecular rulers that I showed you earlier going through the glass nanopore with free translocation and going through with controlled translocation. First of all, we have better signal to noise ratio, but we also have a uniform spacing between the molecules, because in free translocation, you never know how fast it goes through. So there's a lot of uncertainty in the spatial direction. The other advantage is that we now control the speed. So we can translate the molecule as fast or as slow as we want. And the slower we go, the better our signal to noise ratio, the more we can average. So here is free translocation speed at around 100 micrometer per second of a molecule going through. Now we can slow it down, and you already see here qualitatively the improvement in the signal to noise ratio, which also leads to a better precision of the controlled translocation versus the free translocation. The next advantage of this technique is that the molecule is not gone once it's translocated. It's still there, it's still tethered to the surface, and so we can measure it as many times as we want to. So we can go up and down on the same molecule as many times as we want. And this is important because all of these single molecule techniques that are relying on statistics to get a very high resolution assume that each molecule is identical as it passes through a nanopore. Now here we show an experiment where we are doing 100 different molecules, but these molecules are supposed to be identical as far as DNA is identical. You see that there is, yes, it helps to average, but there are a number of different variations because not all molecules actually are the same. And here we're doing 100 readings of the exact same molecule, and you see that the resolution is much, much better, and we're starting to see details in the measurements, such as these changes here that would have been lost if we had averaged over multiple molecules. And now we wanted to push that to see how we can make use of the higher resolution and the better sensitivity. And instead of working with these DNA rulers, which are biologically not very important, we decided to move to DNA gaps. So here, instead of having an additional exclusion in our nanopore, we have less of an exclusion here in the gap region. So we have double-stranded DNA here, double-stranded DNA here, and here we have single-stranded DNA gap. So now the peaks actually go in the other direction, because when you get to a gap region, you have less obstruction, so the current goes up, but it's very well visible. And here is an example of really measuring this gap size here very many times. There's 1,000 readings on the same molecule, and we can average and we can get really very high resolution with that, but we didn't quite know yet how high. So we decided to make multiple of these test molecules, where we had molecules that had 80 base gaps, 40, 20, and 12. And this here are really the raw data curves. And you see we have a very strong signal at 80 base gaps, 40, 20, and even down to 12 base gaps, we still have a lot of signal to noise left. And so we thought, well, how far down can we go? Can we go down to actual single base gap resolution? In order to do that, we made a trick. We used a bigger gap. We used one of the 80 base pair gaps, the 80 base gaps, and hybridized to it the complementary strand that had 79 bases. So that left a single base gap. And indeed, so this is the curves that we get when we're measuring just the bare gap region with 80 base gaps, so a very strong signal. But even with a single base gap, we still very clearly can see the one base missing. And so we really can say that we have a single base gap sensitivity. And the nice thing is that we can actually do this now in a repetitive fashion. So we can do the measurement first with the bare strand, then we can add the complementary strand and really see that it's really a single base gap that we're measuring. So it's a very new technique. If you're interested in it, we put it on bioRxiv, and we're currently in revisions of this manuscript at Nature Nanotechnology. I hope it's going to be out soon. That brings me to the end of my talk, and I'm sorry that I have over talked. I'm not going to conclude much except for that scanning probe microscopy is super exciting. I think it has a lot of potential in biology. It's untapped potential still, and there's lots of different areas where I would encourage people, especially the younger students in the audience, if this is a direction that you're interested in going, it's a great playground. There's a lot of cool things that can be done, and it's a great interplay between life sciences, single molecule, biophysics, and engineering. And with that, I want to thank my team, particularly the people working on this, which were Samuel, Vitautas, Barney, Santiago. Adrian is no longer in the lab, but also my collaborators, Professor Alexander Radinovic, Professor Bastings, and Professor Guernsey, has been doing the biology and working with us on applying our technologies to biology. And then I want to thank everybody for organizing this. Particularly, I want to thank Gustavo, who is spending a year in my lab. It's a great pleasure having him here. And I hope that from this interaction, we will have many opportunities to collaborate in the future. And with that, if you have any questions, I'm more than happy to answer them now, or you're free to send me emails, and I'm happy to respond that way as well.
Thank you, Georg. It's been a pleasure and a full delight listening to your talk. It is really impressive work all the way through. So thank you again for coming and for presenting us with this opportunity. Well, that said, yes, we are a little bit advanced in time, but I think we do have time for a few questions. And does anyone wants to ask away the first question?
Hello, Georg, can you hear me?
Yes, Roberta, I can hear you.
I have a question. These self-sensing cantilevers, are they much more expensive because you are building this electronics in it?
So yes, they are more expensive than a very normal dumb cantilever. But honestly, this is all a matter of scale. Even with normal cantilevers, let's say quasi-normal ones that are very small and made for high-speed AFM imaging, they are around $100 per cantilever. And the self-sensing cantilevers, they're around the same ballpark. So yes, they are more expensive than your garden variety $25 cantilever that you buy for general purpose AFM, but it's not nearly as expensive as some of the other passive cantilevers that people use in semiconductor industry, which can be hundreds or thousands of dollars.
I was thinking if you could construct this electronic separately and then make some kind of coupling, so you could reuse this electronic self-sensing. It would not be self-sensing, but yeah, if you couple both together.
Yeah, one could think about that. It's I see your point, and I think there have been in the past some attempts to do like some hybrids configurations. For us, the key really is, or the challenge is, to bring as much additional capability into the cantilever as possible. So, and maybe this is a little bit a luxury that we have. We don't really care too much about the price of the device. We care about the ultimate performance and that it can do something that's different from what else was able to do. I personally think for normal AFM measurements, people should just use the optical lever detection. It's very, very good. Cantilevers are cheap and it does a great job for that. But there's many things that the optical detected cantilevers just cannot do. And that's where it makes sense to start to put the extra effort in. And there, the extra price in making the cantilevers is not the prohibitive factor.
Okay, thank you. I have another question, but I don't want to monopolize you.
Feel free, Gilberto, to ask your questions. I mean, you're our most accomplished expert in the field, so.
Yeah, the question is, how much can you improve this single base pair detection, improving the shape or, yeah, the morphology of the pore?
Yeah, so this is a very, very good question. That clearly is the next big thing that we have to work on. And there's a little bit of converging of technologies here. It's clearly the biological nanopores are by far the most sensitive ones. And with molecular engineering, people have been able to tune the sizes of the pores very, very well, and even puts some biological specificity in them. But the problem is that through the biological pores, you either have the free translocation, or what people actually do is they couple the biological pores with biological motors that then feed the molecule through it. But there's a lot of stochastics in there in that process. If you now were able to couple a biological pore at the end of such a capillary, then you would have the best of both worlds. You would have the biological sensitivity of the single nanopore, which is exquisite, but you have the direct control of the translocation with the scanning ionoconductance microscope. And that's what we're currently working on.
Yeah, thank you. Congratulations for the amazing work.
Thank you. Much appreciated.
Anyone else want to please present your questions to?
Hi,
You
Georg.
are muted, Pedro.
I have too many buttons.
Georg?
Pedro, você está mutado.
Yeah, okay. So who's talking right now? Please open your cameras if you're yes, thank you, Gustavo.
Sou eu.
Okay.
Hi, Georg. Amazing talk. Please, you talk about the high-speed systems and the workshop to assembling the system. In general, how much it costs to have a system to bring to here, to the institute?
Well, I mean, as you know, the system consists of individual parts, so you don't have to do everything at once.
Mm-hmm.
The AFM head, so the high-speed head, is around 7,000. The high-speed AFM controller, which I did not talk about here today, is around 7,000 plus another 7,000 of electronics that you have to buy from National Instruments if you don't already have it. And then the high-speed scanner is around another 4,000 in material. The reason why I'm saying roughly is because it very much depends on what the prices of the raw materials are at this point. As you know, we have a chip crisis, and so unfortunately prices of electronics have gone up quite drastically. But I'm confident that stuff will settle down. So I think you'll be at around 25,000–30,000 for a full high-speed AFM.
Yeah, because it's really important to talk about this, because if you look for the price of your system and for a commercial one, high speed, it's like 20 times more than this, you know.
But we're not actually doing it just for the price. We're really doing it because we think it's a good way
it's important
to share the technology quickly.
Yes.
Companies are doing a great job building very, very good microscopes for general purposes, but they're always behind in the technology. They have to be. They must be. They're making tools must be maintained and work for everyone.
Yes. Thank you.
Hello, Georg. Just to say hello to you, and thanks for our nice talk. It was very, very
Thank you. Good to see you again.
interesting. How things are going there? It's okay.
It's good. I mean, we are all coming back from the COVID times, and people are back in the lab, and I have the feeling we're looking at a very productive time in the next 1 or 2 years. There's lots of stuff that people were doing, ideas people had during the lockdown and during the crisis of things not being able to be shipped. But now things are coming back, so I'm quite optimistic. It's doing pretty well. You should come by next time you're in Europe. You should come by.
Here also.
We would love to have you.
Solo.
Okay, thanks, Paulo. I believe Professor Fabio Mendonça Gomes wants to ask a question too. So please, Fabio.
Okay, so I was first, congratulations for this fantastic talk. I was very, very impressed when you were showing the build-up of the centriole-like structures. At the same time, I was now asking myself, how much further could you think you could push that in terms of looking into smaller binding partners or smaller structures? And I'm not even sure whether size is the most important component here, or whether there would be something else apart from size that would limit how much further you could push that into the general structure of macromolecules.
Okay. The answer to the question is, how many hours do we have for this discussion?
Can you sum up?
Yeah, I will sum it up. So I mean, it's a very complex thing, and certain things are relatively easy to do, which is experiments like the DNA that I was showing on flat mica surfaces. They're very well suited for AFM. The DNA is negatively charged, mica is negatively charged. We can titrate the adhesion between the molecule and the surface very well. It becomes a lot more challenging when we're trying to make the systems that we are looking at more complex and more biologically relevant. So clearly, an important aspect is doing measurements on membranes. So doing the assembly on mica is nice, but cells don't consist of mica, cells consist of lipids. And so many interactions, many proteins really only make sense to study when you're doing it on a lipid layer. And it is possible, but it does create more challenges. It creates more opportunities also because you can functionalize the head groups of the lipids to specific interactions, but it makes the whole thing slightly more difficult. And then the other issue is studying molecules that function, that do their work inside cells, not on the surface. If the job of molecules is to function in free solution, then anything we do with high-speed AFM is kind of is not that relevant because we have to have them on the surface. Now, what helps us, fortunately, is that a lot of function of molecules does happen on surfaces. It can be the cell surface, it can be the surface of organelles. I mean, the cells are packed, there's lots and lots of stuff inside, so almost everything is an interface. So the biggest challenge is really how to prepare the interface in a way that we can have it nice and flat on the surface and still maintain its biological relevance for the experiment. So we can go arbitrarily more complex, but it requires increasingly better students, smarter students, more motivated students, and that's all that it depends on, right? I'm grateful that you congratulate me for the work, but in all reality, the work is not mine. The work is done by the students and postdocs. We all know that I get the privilege to just present it here, but the congratulations really should go to them.
All right, okay, some work ahead, and congratulations also to the students and postdocs working on that. Thank you.
Okay, so that's amazing. Georg, I believe we can start heading to the end of the talk. Thank you again for your amazing work and the amazing work of your students, which, by the way, well, it's true for all of us in academia. We all know that. We just need to convince our governments that that's true. But yeah, main part of the work is done by students anywhere in the world.
And I know that you have excellent students because, for example, Samuel here, who did all of the scanning and conductance microscopy and single molecule force spectroscopy work, is someone that I recruited from your institution.
That's amazing.
So it's very, very good to have your people over here.
That's amazing. Well, thank you again, Georg. We here in the Institute feel very lucky for having you, and we see all of you next Wednesday, same time, same place. And yeah, bye-bye. See you all.
Thank you. Bye, everyone.
Bye-bye.
Okay, so we now are not live anymore.